plan for today is to start talking about microscopic neuroanatomy. We're going to talk about the elements within the nervous system, the parts of the nervous system, basic components of the nervous system, and this will be in chapter 2. Okay, chapter 2. Cellular constituents of the nervous system, I think that's what it is. Yeah. A quick word about the exams. I don't know 50% of what's in this book, okay? I'm not an expert on neurophysiology. I just got sucked into teaching it because nobody else would. It's a pain in the butt. So, I know just enough to be dangerous, to get by, to be able to pass the board, and to be able to prepare you for your neuro classes, your neuro treatment classes to come. Okay? So you can look through this book and find millions and millions of words. I have no idea what they mean, and I don't care what they mean. Okay? So when you read chapter 2, chapter 10, whatever it is, what corresponds to my notes, don't think you have to know everything in that chapter that I haven't mentioned. Okay? I mean, it's very nice. You should. It's nice to read that and know more than obviously than I teach. But there's no way we could get through all of this in one class. So I pick out what I think the most relevant clinical stuff in there, from my experience treating patients and my knowledge of what you're going to have to face both in the boards and in your neuromuscular classes to come. Okay? So don't go in there thinking I've got to memorize every single word in this book. Just to reinforce hopefully what I'm saying in class. Fair enough? Okay, so we're going to talk about the elements of the nervous system. Before that, we're going to finish off talking about this correlation between neuroanatomy, structure and function. Any questions about anything I mentioned last class? All common sense, right? Basically, if you know your neuroanatomy, you can help understand why those symptoms are presented in this patient with a lesion here. Okay? Which side is proprioception broken the cord? Ipsilateral or contralateral? Some said ipsilateral, some said contralateral. Which is it? Ipsilateral. Proprioceptive. We get to these pathways, obviously in more detail. Basically, they very quickly, quick review. What I try to do, begin the class, is a very, very quickly overview what we did. Some of the key aspects of the previous class: brain, brain stem, spinal cord. Okay? Your proprioceptive pathways, for example, your Golgi tendons, your muscle spindles, etc., they're going to come in without synapsing. They're going to go up to the medulla, synapse, cross over here in the lower brainstem. That's when proprioceptive fibers cross over in the bottom of the brainstem. That's when they become contralateral, as it were. So this is my right leg. Remember, it has to get to my left brain. And they cross over here. Then they ascend through the brainstem to a certain part of the brain known as what? What do all my fibers for sensation, whether it's proprioception, pain, temperature, touch, sight, hearing, what do they go through? What is the relay station that all our sensations go through?
For the motor cortex?
That's where the motor neurons come from. Our sensory fibers. Hopefully you'd have heard of this term before, maybe not. The thalamus? You've heard of the thalamus, haven't you? Yes? Yes. That's a relay station. Every sensation going out to the cortices, the sensory cortex, goes through there. Okay? So that's the pathway for proprioception, right? How about, so that's contralateral to the leg, it's ipsilateral within the cord, right? Same side as the right leg, whatever this is. How the pen temperature do what? How do they get up to my left brain, my right foot? They go in here, right? They synapse at the segmental level, and they cross over. Do that. Okay? So your pain and temperature fibers cross over at the segmental level immediately, and they ascend contralaterally in the cord. So you've got this sensory dissociation within the cord. Motor fibers, most of them, obviously we're going to come back to the details of this later, but most of our motor fibers do this. They come down, cross over, like that. Okay? So your motor fibers, motor pathways start on one cortex, end up at the other leg. And they cross over in the medulla here. Now, the axon crosses over and then goes down on the contralateral side after the medulla. Okay? So those are some of the other pathways. Lots more pathways I'll talk about. But those are some of the important ones in helping diagnose a certain lesion. Alright? This neuron, this green one, that comes all the way down the cord, all the way down segmental level, is known as the upper motor neuron. Is that a term you've heard? Do you know the difference between an upper and lower motor neuron? Yes, no? No. So the upper motor neuron basically is the cell that takes the signal from the motor cortex, in this case, down to the L5, S2, C4, whatever it is, the segmental level. And that's known as the upper motor neuron. One long axon. At the segmental level, another neuron takes over and takes that signal out to my quadriceps, or my biceps, or my triceps, or whatever. Okay? And what's that called? That neuron that goes from the spinal cord to my muscle. Upper versus lower. That is your lower motor neuron. Very, very important terminology. You must know that terminology. I know there's PTs in the 6th year who think upper motor neurons go to the arms and lower motor neurons go to the legs. Idiots. There's a few of them. Well, a lot of them are. No, but some people I don't know why they get confused with that. It's daft. You must know that terminology. Very, very, very common questions in your boards, upper and lower motor neuron lesions. Because if you have an upper motor neuron lesion, it's very different than if you get a lower motor neuron lesion. The signs and symptoms you'll see in a patient are very different. And they're the classic signs and symptoms associated with upper motor neuron versus a lower motor neuron disease. And you must know those. So you must know what that terminology is. Okay? So with that knowledge, we can lesion something, we can put a cut in the spinal cord like that, with a heavy section of the cord, and that's obviously known as a Brown-Séquard lesion. It's very, very rare to see a pure Brown-Séquard lesion, but sometimes you do. Gunshots, knife wounds, damage, whatever, you can get that. But often you get some, not a pure Brown-Séquard lesion, but it's a classic lesion, so you'll have questions on the boards about that. Because it's so obvious, black and white, what happens, and you can see what happens here. The green and the black are cut off. The green is our motor neuron, so I have loss of motor control of this muscle over here, and I have loss of proprioception from this limb over here, whatever it is, obviously. But pain and temperature are fine, because they ascend contralateral to the leg. They bypass the lesion. Alternatively, the pain and temperature from the other side, they're coming in here, right? They can't get up. So you lose pain and temperature from a contralateral lesion, contralateral side below the level of the lesion. So all this pain and temperature from all this part of the body here is gone. Or pain and temperature from here is fine. Alright? Simple, right? Listen to it. Okay, so knowledge of our neuroanatomy gives us knowledge of neurology, gives us knowledge of why our symptoms are there, gives us some clue to the type of lesion and how to treat these patients. Okay? The other thing that can help is this on the chart here. This is the time course of the lesion, the time course of the symptoms. Take the patient's history. When did you have these signs and symptoms? How long did they last? Etc. etc. That will give you some clue to what's going wrong with them. Okay. So what it shows here, these lines, in essence, are time from along the bottom, and these are episodes of the signs and symptoms coming on. So this, for example, would be migraine, this list here. Migraine. Symptoms go, and they're gone. And that's it. No residual symptoms. Like maybe perhaps a TIA might look like that as well. What's a TIA? Transient ischemic attack, right? Your vasospasm sometimes will close the blood vessel off, like a very, 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 very mini stroke. Then it's gone, and you're completely better. That's usually a precursor to something worse coming on later. And this one here, the symptoms are getting worse gradually. What about this one here? This one's a classic sign of a disease where you get a bout of symptoms, lasts for a period of time, and then it goes, and you're back to normal. But after a while, you don't go back to normal. You start getting worse, progressively worse. Then you have this is relapsing and remitting symptoms. You know what I mean by that? Relapsing and remitting. Classic sign of MS, of course. This is your classic MS pattern. Sometimes, that's the most common type of MS, this relapsing, remitting form. Sometimes it just gets worse and worse and worse and worse and worse very quickly and they die. That's the worst type to get, of course. D is, you're fine one day, you have something happen to you, and you've got these symptoms forever. You never get any better. That would be a stroke, for example. Well, hopefully we don't get this plateau at the end. Our job as therapists, of course, is to get that plateau down, hopefully. So rather than going like this, you want to try to improve function after the stroke. And that's your job as therapists, to not let them stay at that plateau of symptoms. You want to help them get better. That's basically you get something, it gradually gets worse and worse and worse and worse and worse. No relapses, remissions, just gets worse and worse and worse with time. And that would be what, Parkinson's probably, something like that. Alzheimer's, Parkinson's is sort of E. And the last one there is where it's the same sort of progression, but it's much, much faster progression. Pretty quickly they're going to die. Brain tumor, something like that. So taking a history of the patient's signs and symptoms will give you some clue to perhaps what's going on, what's wrong with them. Okay? So your patient's going to come in and you're going to, if you're for example, primary care, in home health, I know PTs have direct access. You can be the first person to see this patient, the first person in the healthcare system to admit this patient. Hopefully you're not, because it's a massive pain in the ass paperwork. We hope nursing have gone in before you and done the paperwork. But often I'll go in, I'm the very first patient, the therapist, the very first healthcare professional into this lady's home, living by herself. She's got these symptoms, signs and symptoms. It's up to you to say, is this something I can deal with? Or has this patient got cancer? I've got whatever. Do they need to go and see somebody else? So you need to be able to diagnose some signs and symptoms, what's going on with this patient, hopefully. Okay? Sometimes you get neurological referrals that the doctor doesn't know what it is, so they send it to you. Many times I've had referrals for vestibular issues, for example, balance issues. Doctors, the primary care physician doesn't know anything about balance usually. They'll say, bad balance, treat. Evaluate and treat. Up to you, Ben, do what you want, because I know what's going on here. So we'll spend a lot of time later on talking about balance issues. Very, very big part of your practice. Okay, let's kick off. We'll get going on our review of microscopic neuroanatomy, structure and function, what these things do. Okay, and hopefully I said, let me know if I'm going too fast, but you've all done this in ASP some way. You should have done what a neuron is, what it does, the basic components of that. I might add a few things here and there, but hopefully most of this is just a review. Okay, so before we get to the three components of the nervous system we're going to talk about are the neuron, which is basically the functional unit of the nervous system. This is the wire. This is what the nervous system is all about. Is the neuron or the nerve cell? Okay. Don't confuse a nerve with a neuron. Don't say nerve when you mean neuron. A neuron is one nerve cell. A nerve is lots of these things, either motor or sensory or whatever. Okay. So don't say nerve when you mean nerve cell or neuron. Okay. We have Schwann cells. The other components, major components, Schwann cells. Of course, those are the things that produce the myelin sheath in the peripheral nervous system. Okay? Again, these terms are not new to you, hopefully. Myelin sheath, okay? This is the insulator within the peripheral nervous system. What is the insulator within the central nervous system? The oligodendrocytes. You've heard that term, yeah? Just review, hopefully. Oligodendrocytes. Oligodendrocytes means what? What's site? What does site mean? Cell. Dendro, as in rhododendron. Branch, right? Branch. Oligo, as in oligarchy. Ruled by a few or many cells. So oligodendrocytes they sheathe or they wrap around a few cells within the central nervous system. It's not one-to-one ratio, and there's very different things happening in the central nervous system than the peripheral nervous system. Unfortunately, we do not get regrowth of neurons within the central nervous system, unfortunately. We do get regrowth of axons within the peripheral nervous system, if you're lucky. You can regrow axons within the peripheral nervous system. You cannot, by and large, grow them in the central nervous system. There's a lot of research trying to make them grow, trying to figure out why they don't grow. Because if you did, you could grow axons in the nervous system, you could cure stroke, you could cure spinal cord injuries like that. Unfortunately, we can't do that at the moment. And the oligodendrocytes are part of the third component of a nervous system called neuroglial cells. A neuroglial. You've heard that term, neuroglial? Means nerve glue. It's the glue, the supportive stuff within the central nervous system that's not neuron. Stuff that, we'll get to that in a minute. It glues structural support and other things within the central nervous system. Neuroglial cells. So those are three components, broad components of the nervous system. We'll spend most of the time talking about the nerve, the neuron, nerve cell. The other stuff we'll briefly mention. But first, let's look at the neuron itself. Here's a generic neuron. There are three characteristics of a neuron that separate it, distinguish it from all other cells in the body. They're important to us as therapists. The first one is basically they're specialized for conduction. They're excitable and they send a signal down the length of the thing. Many cells are excitable, muscle cells, etc., etc. Other cells in the body are excitable, but they're not designed to send a signal down them. So neurons are designed like little bits of wire as to conduct electricity, essentially, down them. They're the signaling component within the nervous system. Okay? No other cells can do this, really. The other thing that distinguishes neurons from other cells, muscle cells, bone cells, kidney cells, whatever, is that they're very sensitive to O2 deprivation. They do not like having no oxygen. They will die very quickly if you deprive them of blood. Your muscle's fine, it can function anaerobically for ages. You get lactic acid buildup, it hurts, whatever, but you can run a muscle without oxygen. Use it stored glycogen, etc., and you can run a muscle anaerobically. You cannot run nerve cells anaerobically. You try, they die. That is why strokes, brain attacks, you need to get patients immediately to the ER if they have any signs of strokes. and it's new terminology now. A stroke is a brain attack. You should treat it like a heart attack. You don't just sit there and watch it happen. You get them to the ER immediately. You try to prevent loss of brain cells, because without oxygen they're going to die very quickly. There's like about 10 or so seconds of oxygen in the brain at any given time. You deprive the person of oxygen, you can choke people off, they pass out, that's why. Okay? So you need to have oxygen at all times to all nerve cells. With a stroke, the CVA, ischemic stroke or hemorrhagic stroke, whatever, if that's deprived oxygen, 2 parts of the brain, that brain part will die very, very quickly. Okay? And the third, following on from that, nerve cells, neurons, do not have a mitotic apparatus within the cell body. Okay? Nerve cells do not have any mitotic apparatus. Therefore, of course, they cannot have any mitosis. Which means, basically, they cannot reproduce themselves. You cannot bud off new neurons. You kill a neuron, it does not regrow. You cannot regrow another nerve cell, unfortunately. Okay, it's gone. If we could, a lot of you wouldn't have jobs. There'd be half as many people in this room if you could regrow nerve cells or bud off new nerve cells. You wouldn't have Parkinson's disease. You wouldn't have stroke. You wouldn't have spinal cord injury. You wouldn't have multiple sclerosis, etc., etc., etc. Any time a nerve dies, you just grow another one. Thank you very much. I don't care if I have a stroke. I'll grow some more brain. I'll grow some more spinal cord. I'll have some more, big deal. You damage a muscle, you break a bone, you grow more bone, right? Big deal. But imagine if you broke a bone, it was broken for the rest of your life. That's what basically happens to the nerve system. You cut the spinal cord, it's broken for the rest of your life. Okay? And therefore you get signs and symptoms of a broken nervous system, and that's what you treat. Without this problem, if you did have the ability to grow more nerve cells, you'd be treating orthopedic lesions all day long and no neuro stuff at all. So, good for us, bad for patients. There is one place in the nervous system that you actually do regrow nerve cells. A lot of research on this one little place, trying to figure out how come you can regrow them and nobody else can. Can I make other parts of the nervous system like you? Where is that? There's one place in the nervous system you actually regrow nerve cells. The olfactory nerves, smell. They're continually grown. For some reason, we can grow smell neurons, as it were. They die off all the time. Now, there's a lot of research on it, figuring out, can we make the spinal cord like the olfactory nerve? We can't yet, but it's kind of interesting. If we could, then a lot of you would be out of a job. Okay, let's look at this. So those are three sort of characteristics of a neuron you do not find in other cells in the body. Specialized for conduction of signals, very sensitive to O2 deprivation, will die very quickly, and if it does die, you can't grow anymore. Okay, unfortunately. Most parts of the body we can grow more liver cells, we can grow kidney, we can grow muscle, we can grow bone, etc., etc., etc. Alright, so let's just go over the very basic features of a neuron, a nerve cell. This is a generic one. There's components within it. So, the top of this diagram, this tree thing here, these are dendrites, okay? Again, you should probably have all heard of that. This is sometimes known as a dendritic tree. This is the receptive segment of a cell. This part of the neuron is responsible for receiving signals from presynaptic neurons. Do you know what I mean by a presynaptic and a postsynaptic neuron? Yes? It's obvious, isn't it, really? What it sounds like. Presynaptic is the neuron before a synapse, right? And the name changes, of course, depending on what synapse you're referring to. So here's a muscle, let's say. There's a neuron. I'll often draw little neural circuits. That's the cell body. I don't put the dendrites. The dendrites are here, okay? Axon terminals, muscle. That's a cell. That's a one neuron. So here's another neuron. Okay? So this is the presynaptic neuron. This is the postsynaptic neuron, with reference to this synapse. Obvious, right? So this was a presynaptic neuron with reference to this synapse, but it is the postsynaptic neuron with reference to this synapse. Okay, so obviously name changes. So presynaptic and postsynaptic neuron. Generally speaking, if you see the cell body white or circle, not filled in, that's an excitatory neuron. Okay, that's an excitatory neuron. It means, so in this case, if I turn this neuron here, this neuron A, what will happen to this muscle? What will happen to this muscle if I turn that cell A on?
It's going to move the muscle.
It's going to contract the muscle, right? This will be my alpha motor neuron, remember? The alpha motor neuron goes to your cell bodies, to your muscles, right? Yes? Alpha motor neuron term you've heard, yeah? However, if I fill the head in, the cell body of this neuron here, in most texts, by convention, that is an inhibitory interneuron, an inhibitor. Okay? That is an inhibitory neuron. It turns off the neuron it's connected to.
Does that diminish muscle movement, or it just cuts it completely?
Good question. It depends how strong that signal is. It depends how the other neurons influence it. Yes, good. Yeah, it can do both, depending on the level of background activation from other neurons. So what does it do to this neuron, or this muscle? It will tend to inhibit it. It will turn this muscle off. Let's pretend it's black and white. Let's pretend those are the only neurons in this chain, which is never going to happen. Let's pretend it does. This would turn this muscle off, wouldn't it? You excite this neuron, and you turn this muscle off. What about if you did this? I am going to excite cell B. What happens to the muscle?
Well, still nothing. It's not connected to the muscle, is it?
There's a circuit. If I excite cell B, it inhibits, this is an inhibitory neuron, right? Inhibits the inhibitor, which means you excite the muscle. Does that make sense? If you inhibit the inhibitor, that's equivalent to facilitation. Okay? This is your pain gating theory, right? All that kind of stuff. You inhibit the inhibitor, or you inhibitory interneurons, etc., etc. We'll get to this later, but just want to be aware of this kind of circuitry, or this kind of convention when we discuss neurons or draw circuit diagrams. If you see the head of the cell body filled in, it's usually most texts will be inhibitory neuron. We have these inhibitory neurons sitting in the spinal cord all the time. Okay? So, presynaptic, postsynaptic neuron, you know what that means? Okay, let's get back to our neuron. So the dendritic tree is the receptive portion of the cell. It's responsible for receiving all those signals, for example, here, on the top left corner there, that is a cell body, the dendrites, and all those axon terminals synapsing on it. So all these axon terminals going on to this postsynaptic neuron here. So again, it's the receptive portion of the cell. Okay? Within the dendrites, we have what's known as decremental conduction. Familiar with that term? Yes? No? Yes. Pardon?
Need a refresher.
You need a refresher. Just you. Anybody else need a refresher? I mean, just you, I won't bother, but if anybody else needs a refresher, I will give you a quick refresher. Decremental conduction basically means decaying. It dies off. Within the axon, of course, what kind of conduction do we have? All or none, right? If I send a signal down the axon, it's going to go to the end without stopping at the same speed, the same amplitude, all or none. It either fires or it doesn't, right? An axon. We'll talk about signaling later briefly, but an axon, we do not get, well, I shouldn't say that. We get this all or none firing. But within a dendrite, we get decremental conduction. So here's my cell body, let's say. Here's a couple of few dendrites, whatever. So if I'm going to send a signal to this is my presynaptic neuron, I want to cause the next neuron to fire, right? My job is to cause the next one to fire. So I send a signal into the dendrite and I cause an electrical current to flow towards the axon hillock. But it decays in strength the further it gets away from where it's input. So the signal strength is going to gradually decay to get to nothing. It's not all or none. It decays. Okay? You, as a presynaptic neuron, want to cause that neuron to fire, usually if you excite a neuron. You want to get to this thing here, don't you? This is the axon hillock. That's the initial segment of the neuron, sometimes called. That is the integrative center. It integrates all the signals impinging on it from various other presynaptic neurons. This is the thing that causes the neuron to fire or not. It weighs all the signals coming from all of these axon terminals, all the dendrites are all going to go through, so you've got this decremental conduction going through the dendrites, going through the cell body, trying to get to the axon hillock. If the axon hillock adds up, constantly integrates, and I've got a bit of strength, I've got a bit of strength, yes, that's enough, I'm going to fire. Once you get above threshold here, it fires it all or none down the axon. But you've got to get enough signal strength to the axon hillock to be able to get above threshold, to get at a certain level, to be able to fire the neuron. If I'm a long way away from the axon hillock, my signal is probably going to die before it ever gets there. So you get this temporal and spatial summation. You've heard those terms? Temporal and spatial summation. So if this one fires a lot quickly, then it adds up and gets more signal strength. And it's got less likely it's going to die before it gets to the axon hillock. Temporal and spatial summation? Yeah. So temporal summation is basically, rather than this, I'm a presynaptic neuron, rather than just firing it once and a second, 2 seconds later I fire again, and another 3 seconds later I fire again, I fire now and I fire now and I now and now, I keep firing lots of time, very, very quickly. And what happens, those signals add up. They build upon each other because they're so close within time, temporal summation, they add up. And you're getting more of a signal going along the dendrite. Okay? It's like shooting a bullet out of a pistol. I shoot it 10 times faster. And more bullets hit that target faster. And there's more signal getting there. So they add up in time.
So it's more likely to reach the...
It's more likely to reach the axon hillock with enough strength to fire, yes. Okay, so that's temporal summation. Spatial summation is, okay, so there's my signal from this fella here gradually dying off. But if I'm sitting here, another synapse, I can add to that in space. So as 2 or more axon terminals add up because they're close together in space. So if you have more than one firing from different places on the dendrites, signal strength is going to add up and you're going to get more chance of firing the axon hillock. Okay?
What was that called again?
Spatial summation. So you've got temporal and spatial, time and space, in essence. Makes sense so far? So all these axons, these terminals, all these are trying to fire, send their signal strength over to the axon hillock. The closer to the axon hillock you are, the better chance you're going to influence that axon, right? So there's a lot of competition to get close to the axon hillock. You also find inhibitory interneurons. Those black dots we talked about, those are trying to turn off the axon hillock. They're going to send negative sort of signals to the axon hillock. The facilitator sending positive signals. One says activate, one says shut up. Don't do anything, go to sleep, don't fire. So these pluses and minuses are fighting to get the axon hillock to fire. And the axon hillock says, oh, I've got 10 pluses and 2 minuses, I'm firing. On the other hand, oh, I've got 10 pluses and 20 minuses hitting me, I'm not going to fire. And you'll find your inhibitory interneurons generally are closer to the axon hillock. They're the final gateway to allowing the axon to fire. So they will fight to get to the close to the axon hillock. So they have more influence. They've got more chance of stopping it if they're closer to it. So the influences are more important. More important. You turn off a neuron using these inhibitory interneurons trying to shut the axon hillock down, as it were. Does that make any sense? So this is all the interesting things happening. This is where motor learning occurs. This is where change in the nervous system occurs. These regrowth of these axon terminals onto the synapse. It's this synapse is where all the interesting stuff occurs. Your learning, etc., etc. Okay? So, on the way to, we've kind of skipped a section, on the way to the axon hillock, we get our cell body. This is our trophic segment. We have a receptive segment, our initial segment, or integrative segment. Here we have our trophic segment. This is the bit that nourishes the rest of the cell. So all the usual cellular stuff occurs. Sometimes called a perikaryon. You heard that term? Or soma. All names for the same thing, the cell body. Okay? It has the nucleus and all the general stuff you'd find in any generic cell. Okay? But it does not have a mitotic apparatus, like other cells. So you do not have mitosis. You cannot regrow nerve cells. Okay? They do have very, sort of slightly distinguishing from many different types of cell. We have our nissl bodies. The nissl bodies are very prominent within, you find these in other cells, but they're very, very prominent within nerve cells. Franz Nissl was a German neuroanatomist, born in 1860, but trivia for you. And these were named after him. What do they do? What do nissl bodies do in, you did cell biology, right? What did you talk about? What do they do? They produce microtubules, little pipes. Okay? Which basically form two functions. They provide a skeleton, the internal skeleton for nerve cells. The internal skeleton gives it the structure, the stability, particularly in the cell body. But more importantly, these tubes form these thingamajigs here. These neurofibrils. Neurofibrils are basically long pipes that go down the axon, which make up the axon really. The axon is bundles and bundles of these microtubules. In that case they're called neurofibrils. and they are responsible for transporting stuff, not the nerve signal, but stuff up and down the axon. Nutrients, enzymes, mitochondria, neurotransmitters, etc., etc. Okay? So obviously we want to get our nutrients down to the axon terminals, we want to get them down to the end. The axon terminals down the far end here don't produce neurotransmitters, they're made up in the cell body. And they're sent down these microtubules, these neurofibrils, to get to the axon terminals for release at the synapse. Okay? Fair enough. So those are very prominent, very important within a nerve cell. Let's look at the axon itself. This is this long wire, as it were, that comes away from the axon hillock and goes down and terminates at axon terminals or telodendria. How long is an axon? Varies from what to what? What's the range of axon length?
Up to a meter.
Yeah, at least easily a meter long, less than a millimeter, from less than a millimeter to this long. Basically, it goes from the top of my head down to the segmental level. Well, proprioceptive neuron, it's a unipolar neuron, we'll talk about later, but a proprioceptive neuron basically can start in my ankle and end up in my medulla. So from my neck, basically, down to my foot. That, in essence, can be one axon. So very long and also very, very, very short. This is the conductive segment of a neuron. This is the electrical wire. This is the thing what does all the work. It sends the signal down, etc., etc. The outer layer is the axolemma. Where is it on there? Yeah, this term down here. This is the axolemma. The outer layer surrounding all that is the axolemma. Okay, and then down the middle of this, of course, are neurofibrils that are used for transport. And there's two ways we transport stuff, not the nerve signal now, I'm talking about. There's two ways we transport stuff down, up and down an axon. One is known as axonal transport. So there's two ways we have to send stuff up and down an axon, nutrients, waste products, etc., etc. We have axonal transport and we have axoplasmic flow. Axoplasmic flow and axonal transport. Two ways we can get stuff up and down the axon. Axoplasmic flow is a slow process. It's basically the protoplasm just stream down like toothpaste getting squirted down a pipe. Streams down the axon going down to the other end, essentially. This is a slow process, but it takes big stuff like big enzymes, proteins, etc., can go down, and we'll touch on this in more detail when we look at something with the nervous system. It's a one-way system, though. Only goes proximal to distal. That's axoplasmic flow. Axonal transport, though, is the function of these microtubules. That's fast, and it's both ways. If we want to get something quick down to the end, we use axonal transport. Very fast, through the microtubules. It's a very active process. So we send neurotransmitters, we send things down quickly to the end, and things come back up. Like, waste products, or, the axon terminal are continually sampling the environment, like nerve growth factor, for example. The axon terminal are sniffing the synapses constantly, and they'll sense nerve growth factor release, and they'll bring those back up into the cell body through axonal transport. Unfortunately, bad things can come back up. That's how infection things can get into the central nervous system through axonal transport. coming from the skin, from a sensory receptor back into the central nervous system. Like rabies, for example. That's how it gets in the central nervous system. It goes up through the nerves, through axonal transport, quickly. Alright, last part of the nerves neuron, these axon terminals down the end here. This is where the synapses, of course, these telodendria, we have these synaptic end bulbs, which release these vesicles, these little capsules, these synaptic vesicles, release these neurotransmitters into the synaptic cleft. Okay? And their job is to, excite to open the channels, the ion channels in the postsynaptic membrane, try to make the next neuron fire in essence, or inhibitory. There's a little gap there, there's a delay, so it's nerve transmission down the axon is very, very fast. Maybe hundreds of miles an hour in some cases. We'll look at the thickness and all that kind of later. But in fact, there is a gap, there is a gap there. 200 angstroms wide, typically. What's an angstrom for your bit of trivia, for your next time in your pub trivia game? An angstrom is 100 millionth of a centimetre. So it's pretty small. But it's slow, because while there's an electrical signal flying down here very rapidly, this is diffusion. There's diffusion of that neurotransmitter across the synaptic cleft, and there's obviously a delay there. So things slow down there. And we can have excitatory neurotransmitters released, or we can have inhibitory neurotransmitters released into that synaptic cleft that try to either excite or inhibit the postsynaptic neuron. What is the most common excitatory neurotransmitter? Let's take a few minutes to talk about neurotransmitters while we're at the synaptic cleft. What's the most common organ excitatory neurotransmitter you've heard of? Acetylcholine, right? Acetylcholine is an excitatory neurotransmitter. Where do you find that? In what nerve cells release acetylcholine, or acetyl, how do we pronounce it? Well, the motor neurons when it's firing a muscle, okay? The somatic nervous system. You know the difference between somatic and autonomic, yes? Somatic and autonomic nervous system. So somatic nervous system going to my biceps is going to release acetylcholine to drive that muscle, to cause that muscle to contract, okay? So let's pretend I've released it into this synaptic cleft here onto my muscle. Does it stay around there forever? No. What happens? What happens to our neurotransmitters once they've been released? Very shortly after, they are reabsorbed, they're broken down. And what's the thing that breaks down acetylcholine? Cholinesterase. You've all heard that, right? Cholinesterase is an esterase that breaks down acetylcholine, prevents it hanging around in the synaptic cleft for days or whatever. Okay? Fibers that release acetylcholine, of course, are called cholinergic fibers. Cholinergic fibers or cholinergic neurons release acetylcholine into the synaptic cleft, turns the muscle on, whatever it is, and then you get cholinesterase coming along to break it down, and so it's not there forever. Have you heard of this drug? Neostigmine. Anybody heard of that? Any uncle, granddad, father taking neostigmine? Neostigmine is a cholinesterase inhibitor. It's trying to stop cholinesterase working, inhibits it. And why Why the heck would you want to do that? Why would you want to stop the thing that breaks acetylcholine down? Why would you want to stop it working? What patients would you think would benefit from getting an injection of that stuff? No, I mean the issue with Parkinson's, what's the neurotransmitter most problematic in Parkinson's?
Dopamine?
Dopamine. Good thought, but no, dopamine is a whole different issue. We give people L-Dopa, a precursor to dopamine for Parkinson's. Well, who has, okay, there's a disease, particularly affects the cranial nerves, particular face, in which there are antibodies sitting on these receptors here, acetylcholine receptors. Okay, so you've got these antibodies that sit on the acetylcholine receptors, and therefore the acetylcholine has less chance of activating them, right? Makes sense? So if you've got 50 receptor sites, let's pretend, for acetylcholine on this membrane, and there's antibodies sitting on 20 of them, or 40 of them, you've got 10 left. So you want acetylcholine to hang out in that synaptic cleft as long as possible. You ain't broken down because you want to make sure you get to those 10 active open receptors, right? Makes sense? So if you've got antibodies blocking acetylcholine receptors, you want to stop cholinesterase working to give the acetylcholine the best chance of getting to those open receptors. It's exactly why you give this drug to somebody who's got what? Myasthenia gravis. Have you heard that? Myasthenia gravis? We'll talk about this in detail later, but so I said, we start a clinical course, but when I come across something interesting, relates, I'll mention some clinical aspect just to relieve my boredom. And it's more interesting, obviously, like blah, 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 blah. Myasthenia gravis means myo, muscle, sthenia, weakness, gravis, grave. Grave muscle weakness. Nobody seen, has anybody heard of myasthenia gravis? It's not all that uncommon. We may often, somebody's mum or dad has got myasthenia gravis. It's basically a disease that we'll get to it later, but it basically attacks the synaptic junction, right? And it will affect the bulbar nuclei first. In other words, the cranial nerves. It attacks cranial nerves first. The muscles, it lies on the muscles that cranial nerves go to. So, a typical patient with myasthenia gravis will complain, they'll get weak facial muscles, particularly at the end of the day. They'll drool, the eye will, the ptosis of the eye, the face will change, the facial muscles basically get very, very tired and weak, particularly towards the end of the day. And in severe cases, you'll give them some neostigmine. What cranial nerve you really don't want it to affect? What's going to kill you? The vagal nerve. If it gets into the vagal nerve, you're in a mess. Okay? Or even some of the muscles for swallowing. You can choke, aspirate, things like this. Gag reflex can go. Because in severe cases it can affect the ability to swallow or reflexive closure of the epiglottis, all this kind of stuff. So it can get pretty serious, but it's usually a long-lasting disease. And I've had quite a few students who had their mums had myasthenia gravis for many years. So what you do in severe cases, they'll carry an injectable pen and jab themselves with neostigmine if they're in respiratory distress, something like this. And that's what it does. It blocks cholinesterase, so you have more acetylcholine within that synaptic cleft. Let's look at a few more neurotransmitters while we're at it. Since somebody mentioned dopamine, dopamine is made primarily by the substantia nigra. You've heard of that, yes or no? Substantia nigra means what?
Black substance.
Yeah, black substance, which it comes from. There's a lot of melanin producing cells, they're staining cells within there. The substantia nigra is deep within the brain, deep down under the cerebrum. We'll get to this later. But basically it produces dopamine, the substantia nigra. And that is sent through the nigrostriatal system. It's on there. Dopamine. Yeah, the nigrostriatal system, this word here. So it's sent throughout the center of the brain using axonal transport for other neurons to use dopamine. What happens with Parkinson's disease, for some reason, we don't know, the substantia nigra degenerates, dies off. And when it gets critical, I think it's like 70-80% of cells have to die before you start seeing symptoms in your patient. So you've got loss of dopamine, which affects the basal ganglia, and you starting to see these signs and symptoms of a patient with Parkinson's disease. What's the first symptom you see in Parkinson's disease patient, perhaps? This shaking like this, right? Pill rolling tremor, that's sometimes called. Looks like I've got a pill between their fingers and they're doing this. They're rolling the pill in their thing. And so they'll hold on to something like this, stop it doing it. They want to, aesthetically pleasing, so they'll hide their hands, something like this. Okay? Pill rolling tremor. And then what do you get? What are the symptoms you see in Parkinson's disease? Gait disturbances, very short, shuffling gait. We'll get to this later, but they'll sort of walk like this, shuffling. They'll freeze, episodes of freezing. They'll stand still and can't move. Their feet are stuck to the floor, like glued to the floor, and they shake and shake and shake and try to get the foot off the ground. They can't. Particularly in doorways and things like that, freeze. How many people know somebody with Parkinson's disease? Quite a few. They're quite common. And we talk about signs, symptoms, and maybe a bit of treatment for this in motor control class. Everything gets smaller. They get micrographia. What would that be? Micrographia. The handwriting gets very, very small. Very small writing. The face gets very blank, expressionless. Everything they do gets smaller. They speak quietly and whatever. Lots of things we can talk about in terms of treatment of this. But the most common treatment is we give them L-Dopa. You can't give somebody a squirt of dopamine because it won't cross the blood-brain barrier. This is a precursor to dopamine. It does cross the blood-brain barrier, is made into dopamine within the brain and then uses it. It's just some amazing videos of people on/off meds. You see them off meds and they're like, can hardly move. You stick this stuff in, half an hour later, you wouldn't know they've got Parkinson's disease. Okay? But you only got, you have this for 5-10 years, you start getting dyskinesia, all kind of stuff, this sort of side effects of this, and it wears off, etc., etc. So there's a lot of stuff looking at deep brain stimulation, all the kind of techniques we'll talk about that people apply to treat Parkinson's disease. Let's go through a couple more. Epinephrine and norepinephrine. Epinephrine is produced, or sometimes called, epinephrine is also called what? Adrenaline. Or norepinephrine is noradrenaline. Epinephrine, of course, made in our adrenal glands. Norepinephrine or noradrenaline. Very, very important in what part of the nervous system is noradrenaline or norepinephrine used. Where do we find that? Autonomic nervous system, right? Autonomic nervous system. What neurons within the autonomic nervous system release norepinephrine? Two parts of the autonomic nervous system, right? Parasympathetic and sympathetic. What's the parasympathetic do? Rest and digest. Sympathetic, flight or fight. Okay? Within each of those systems, you have a chain of neurons, right? Pre-ganglionic and post-ganglionic neurons, if you heard that term? Yes, no? Refresher or not? Silly question, huh? This is the somatic nervous system. Muscle, spinal cord. Okay? Somatic nervous system activates skeletal muscles, biceps. There is one neuron, starts in the cord and comes out to the muscle. There's millions of them, but in series there's only one. This is our alpha motor neuron, okay? One of them. I mean, one in series. This is an organ or a muscle within the autonomic nervous system. We have a chain of two, don't we? Not one, but two. A chain of two sequentially get the effector. The sweat gland, the smooth muscle of my arches, whatever. And these are known as what? Pre-ganglionic and post-ganglionic. Because somewhere there's a ganglia swelling that contains lots of these cell bodies. Okay, so remember, all our autonomic nervous system goes through a ganglion. There's various places where this will be, and we'll talk about when we get to the autonomic. But basically we have a pre-ganglionic and a post-ganglionic neuron in a sequence in the autonomic nervous system. Whether it be parasympathetic or sympathetic, we always have a chain of two. So my question again is, where is noradrenaline release? Let's pretend this is the parasympathetic nervous system and this is the sympathetic nervous system chain. This is pre and post. Why here? What do parasympathetic neurons, pre-ganglionic parasympathetic neurons release here? What is the neurotransmitter released there? Any idea? You've done the autonomic nervous system in AMP, right? You just forgot. You were taught this. This is elementary school stuff. Well, high school stuff. This is acetylcholine is released there. What about post-ganglionic neurons? What do the post-ganglionic neurons within the parasympathetic nervous system release here? Acetylcholine. Alright, sympathetic nervous system chain. Pre-ganglionic neurons release what in its here? Acetylcholine. What do post-ganglionic neurons in the sympathetic system release? What are we talking about?
Yes.
Noradrenaline. That is where we find our post-ganglionic sympathetic nervous system releases noradrenaline, norepinephrine. Let's say right there, somewhere. Sympathetic nervous system. Okay? This is the butterflies in the stomach. That's what you feel, and you get all excited and you're scared. That's what's being released. That noradrenaline is flooding the system, and you're all anxious and blah 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 blah. Right? Except sweat glands. Sweat glands. If this is going to sweat glands, it's not noradrenaline, it's acetylcholine. Okay? Okay. And we'll touch on these neurotransmitters as we come to various clinical issues we've got involved in them. Serotonin. You've heard of that neurotransmitter? Serotonin, very important in wakefulness, alertness, and mood. Very used a lot in the reticular activating system. Have you heard of that term? Or reticular formation. Reticular formation, yes, no? We'll talk about that in detail later, but it's basically a network of neurons within the brainstem. It keeps us awake. If you destroy the reticular activating system, you go into a coma or you die or sleep or whatever. Okay? Problems with sleepwalking, narcolepsy, all this kind of stuff. Those are issues of the reticular formation. And it's serotonin is one of the principal neurotransmitters within the reticular formation. It's used throughout the brain. It alters moods. So, when I get up in the morning on a Monday morning, I know I have to come and teach this class. I pop myself some Prozac because to prevent myself being clinically depressed. That's what people take. Well, right? You've got a Prozac. It's prescribed to folk who get clinical depression, and what Prozac does, it inhibits the reuptake of serotonin. So if you give somebody Prozac, there's more serotonin hanging around the brain longer, and you feel good about yourself. That's the only way I can come to this class. 3 pills of Prozac on a Monday morning. That'll do me a one. So it's a happy pill. It makes you feel happy, and that's what people on clinical, seriously, people on clinical depression sometimes go on Prozac, and that's the way it acts. It acts on synapses, and it acts on serotonin reuptake, prevents that happening. Most of these neurotransmitters we've been talking about are excitatory. Dopamine is an inhibitory neurotransmitter, but probably the most common inhibitory neurotransmitter is this fella here, GABA. You've probably heard of that one, yes? Gamma-aminobutyric acid is an inhibitory neurotransmitter. That turns things off. Okay, it kind of quietens things down, hopefully. Many of our drugs, so the synapse is where all interesting things happen. Our drug action occurs. Most of our drugs act at the synaptic cleft. Baclofen, have you heard, is a drug you'll come across later on. Baclofen, have any heard that term? Do you work with peds at all? Yeah? What does it do? Who gets baclofen? Often a spastic diplegia, a child with spastic diplegia maybe, with cerebral palsy. It's often used in treatment of spasticity. Because baclofen is a very powerful GABA agonist. What do you mean by GABA agonist? GABA agonist is something that mimics the action of GABA. It's an inhibitor. A GABA antagonist would do something opposite of that, wouldn't it? So acetylcholine antagonist, acetylcholine agonist. It's the opposite or the same thing as acetylcholine. Baclofen, you'll see in children, it's a very powerful GABA agonist, and you have to very, very carefully titrate this. You give them too much, the kid is floppy and falls and sleeps and can't do anything. If you give it properly, you're supposed to be able to reduce spasticity in the legs in various places. They have a baclofen pump. The kid has a pump on their body and drips baclofen into the spinal cord constantly. Try and reduce spasticity. What's the other drug you give kids in CP? Anybody treated anybody with CP? Yes? Nobody's not a clinic much, yes? No?
Botox.
Yeah, Botox. Who else gets Botox besides children? Little Hollywood stars, right? You're looking wrinkled, looking haggard today. You go and squirt some Botox into your face. And that gets rid of the wrinkles because it kills motor end plates temporarily. If few months later you have to go another squirt into your face. It basically kills the motor end plate at the synaptic junction, prevents that muscle contracting, prevents the wrinkles in your face. Okay? More importantly for us, some of our children will be on Botox. Botox comes from where? What does Botox stand for?
Botulism toxin.
Botulism toxin. It's food poisoning, right? Food poisoning. That's what you give the people in their face. You give them food poisoning to stop them having wrinkles. Okay? And this is a very powerful killer of motor end plates, and children with spastic diplegia who've got very, very hyper-contracted, we'll talk about later, gastrocs, they'll squirt Botox into their muscle to try and kill it, to allow them to get the heel onto ground. It's very painful. I had a child who gave her Botox in the ham. She was 18 years old, and she walked like this. This is how she walked. Because her hamstrings and her gastrocs was tight as knots, and she could not straighten the knee, and she couldn't get her heel down. And so their doctor had given her Botox into the muscle to basically reduce the muscle tone, and she was telling me it hurt like hell. Massive, massive pain from this. And sometimes I question the effectiveness of this, but whatever. We'll talk about later. What time are we supposed to stop? 55? 5 to? Or 50? Yeah, okay. I can talk about one more thing then. Let's pick another one. Glutamate. We're going to come back to all these later. Very briefly, glutamate. Glutamate is a very, very common neurotransmitter. It's found throughout the nervous system. Some of these other neurotransmitters like dopamine and serotonin, we find in certain parts of the central nervous system. But within glutamate, we find it all over the place. Nearly every nerve has receptors for glutamate on it. Glutamate can affect nearly every part of the nervous system. It's all over the place. And it basically it causes calcium to flood into the next cell. So it excites the next cell. And the reason I mention why this is important is because if you have too much glutamate, it causes the next cell to run itself to death, to burn up. Because calcium regulates intracellular metabolism. Remember that from your cell biology, biophysics class, whatever? Calcium drives internal metabolism of the cell. So if you put calcium into a cell, it's going to run itself, churn itself away. So if you have too much calcium or too much glutamate, the cells around this will burn out, rev up and burn themselves to death. Okay? And this is what happens in certain neural lesions, neural insults, like a stroke and spinal cornea particularly. Okay? So it's thought that when you have an insult into the spinal cord, glutamate floods out and causes the neurons around it just to run themselves to death. It's a very powerful excitatory neurotransmitter, and therefore there's some research now trying to prevent this happening, and they'll give them a glutamate antagonist. Glutamate antagonist to try to prevent some of the effects of a stroke or spinal cord injury. So to sum up, synapse, the key part here, all the interesting stuff goes on there. The drug action. What's the most common drug you take to affect your synapses? What's the most common drug you take? Caffeine, right? A cup of coffee, a Coke. It's caffeine. What does that do to you? Is it inhibitory or excitatory? Excitatory, right? It lowers the postsynaptic membrane threshold. It makes the synaptic membrane more likely to fire. You pour caffeine down your neck, you get jittery. Okay? So caffeine, all our drugs. One last drug. I'm walking through the Amazon jungle and I get a blow dart in my neck and I keel over dead. What have I just been hit with? curare. Right, curare? So when you die in the jungle, you know what you've got, right? Just before you keel over dead, you go, oh, that was curare. So those Amazonian Indians stick curare on the end of their blow dart and fly into you, and it blocks acetylcholine. It blocks the neurotransmitters site on muscle. And therefore the monkey keels out of the tree because it can't hold on to the tree. And there's your food. Alright, we'll carry on, finish off neurotransmitters next class and whatever. Talk about something else.